Hello friends, today we are going to discuss one of the important topic about tryptophan operon. Tryptophan operon. In last class we have discussed about lac operon. In that context we have learned about gene regulation. The beauty of gene regulation is the cells, that means the living cells, adapt to the surrounding environments by appropriate molecular regulatory mechanisms. In the sense, say for example, we have around 30,000 to 40,000 genes in a human genome. So for example, a bacteria have some 4,400 genes. Particular example, say we have insulin gene in almost all nucleated cells, but the specialized pancreatic cells called islets of Langerhans only will transcribe the insulin gene. Why not others? That means the gene expression has been facilitated in the islets of Langerhans but has been prohibited in the other cells, though there are insulin genes in there. That means in other nucleated cells. That's the beauty of gene regulation, because gene regulation is very essential for growth, development, proliferation, even for our very survival. So accordingly, there are two concepts, positive regulation and negative regulation. Positive regulation is that regulator element which increases the gene expression, whereas negative regulation means which decreases the gene expression. We have learned about lac operon. It has both positive regulation and negative regulation. Positive regulation means we have catabolite gene activator protein cap coupled with cyclic AMP, which binds to promoter, then the RNA polymerase that leads to transcription. That means transcription is on. Whereas it is also a negative regulator because the repressor protein will prohibit. Of course, we have an inducer, that means the lactose, then which leads to structural conformation of the repressor protein, thereby allowing the transcription. That means it is an inducible operon. That means lac operon is both positive and negative regulation, and the nature of operon is inducible. Okay, and we have discussed about constitutive genes, that means whose expression is unregulated, like enzymes necessary for Krebs cycling, because they are housekeeping genes. They require, the requirement of such genes is sustainable and should be synthesized at a constant pace at almost all nucleated cells. Whereas there are other genes like inducible genes, there is a requirement of molecular signals. I told you, either inducer or repressor. So these are different basic concepts of gene regulation. And today we are going to learn about tryptophan operon. The distinction between the lac operon and the tryptophan operon. The lac operon, I told you, it's both positively and negatively regulated, whereas this tryptophan operon is one of the example for a negative regulation. Because here we have a repressor, I will tell you how it works, which prohibit the gene expression. Whereas in case of lactose operon, there is both positive and negative regulation. Here, and the kind of operon in case of lactose is inducible, but here, that means tryptophan operon is repressible operon, because there is a repressor protein which reduces the expression of genes. That means with respect to tryptophan operon, it is a repressible operon. Repressible operon. At the same time, attenuator operon. I will tell you what exactly it is. And it is a typical example for negative regulation. Negative regulation. That means gene expression is reduced. Gene expression is reduced. These are the basic concepts. Okay, how and what exactly? We have discussed what is operon. It is nothing but a coordinated unit of genetic expression. That means a group of genes acting in a coordinated manner, just like tandem to get the things done. That means the transcription and translation. It's like coordinated fashion. A group of genes acting as a one functional unit is an operon. Say lac operon means lactose is an inducer in that operon. The same, the tryptophan operon means here this operon objective is to synthesize the amino acid, the aromatic amino acid, the tryptophan. That means tryptophan is an aromatic amino acid, which is an essential component in majority of the protein. So synthesis of this amino acid is a main objective of tryptophan operon. How? There are requirement of enzymes, because here we cannot synthesize directly the tryptophan operon. Here there is a intermediate pathway, like say chlorismate. Chlorismate is a substrate that will convert into L-tryptophan. You know, the kind of amino acids we have are L form, dextrorotatory, levorotatory, okay? I'm not going to the details, L-tryptophan. It's not a single step process, it's a phase, like, you know, intermediate phase, like precursor 1 become precursor 2 because of one enzyme, and precursor 2 become another precursor 3 because of another enzyme, and eventually it form L-tryptophan. So that means acting of different intermediaries here, different intermediaries. How? Here by means of 3 enzymes. Therefore, synthesis of these 3 enzymes is the main objective of tryptophan operon. And these enzymes will convert chlorismate, a substrate, into L-tryptophan. That's how the issue ends. That's how this. That means what we call biosynthesis of amino acid tryptophan. It's not a single step process, it's a phase manner, okay? But here we're not going to discuss about biosynthesis of amino acid tryptophan, but how the tryptophan operon regulates, that means how it works. Okay, so the 3 enzymes are necessary for conversion of chlorismate to L-tryptophan, and it's about how the tryptophan operon operates. That is the main objective. Okay, these are the basic component. It is a negative regulator of operon and repressible and attenuator. Okay, regarding its structural aspects, you know, just like lacoperon, it also have 2 components. One, the structural genes. 2, the regulatory elements. Here, unlike lacoperon, here there are 5 structural genes: TRPE, D, C, B, A. Regulatory elements are common, like promoter, operator, which are upstream to these structural genes. In addition to these promoter and operator, there is an attenuator. I told you. Another word I have used, attenuator, more, because it is the second site of controlling the tryptophan operon. It is very important, not only an examination point of view, but in conceptual learning as well. Okay, in the sense, say tryptophan is a structural gene. First we have to draw the tryptophan. It's a promoter, it's an operator, then there is a specialized region, what we call attenuator. Then the structural genes come: E, this is D, this is C, this is B, this is A, and this is terminator, T. That means this is promoter, this is terminator, and these are the structural genes. They synthesize one polycistronic mRNA, you know, one polycistronic mRNA. With respect to all these 5 structural genes, that means 5 structural genes transcribes into one polycistronic mRNA. It is nothing but mRNA that transcribes more than one protein. That means translates more than one protein. This polycistronic mRNA, and it is an exclusive feature of prokaryotes. You know, so one polycistronic mRNA is enough for translation into more than one enzyme. That means here the enzymes. Okay, here the kind of protein is an enzyme. Okay, and this is one operon. But this is not as simple as it is. There are different things and different situations which affects the operational mechanism of tryptophan operon. Sometimes, you know, another mRNA will be found based on last 3 genes. That means C, B, A. So 2 types of polycistronic mRNA. 1 out of 5 to the last CBA. So it's about how then they will subject to the translation process. I told you now in the previous discussion, the enzymes, they will translate, but it is not as simple as it is. It depends on the availability of tryptophan, whether there is a higher adequate, that means adequate amount of tryptophan or not. So we have to categorically explain or discuss for better understanding. You know, for example, there are 2 situations. Okay, there is high or adequate tryptophan. You know what happened? This case 1. You know what happens? I told you already. Tryptophan operon is a repressible operon. What is repressible? Here there is a repressor protein. Here repressor protein reduces the expression of the gene. Here, unlike lacoperon, here repressor protein is weak. What we call apoenzyme. Apoenzyme is it is inactive. There is a requirement to bind with another form, that what we call co-repressor, so that it can attain functional activity. Say this is a repressor protein. Say this is TRPR, which synthesizes the repressor protein. Okay, this is a repressor protein. What we call TRP repressor. But here there is high adequate tryptophan. That means, say, because we require, I told you, tryptophan in synthesis of different proteins, and there is ample availability of tryptophan. So there is no requirement of further tryptophan. That means the expression, the transcription should shut down. How? In general, if there is ample availability of tryptophan. Say these are the tryptophans. These are the tryptophans, and they will bind to the repressor protein, forming a active part. Active part, so that it will binds to the operator. It will binds to the operator. So since if it say, I am only magnifying the operator part. Since it binds to the operator, expression is off. There is no further transcription. That means simply the gene expression is inactivated. This is one mode. That means the tryptophan is a co-repressor. Tryptophan acting as co-repressor, and it binds to the repressor protein, which in turn binds to operator, thereby no RNA polymerase. This is RNA polymerase. Okay, so this is blocked off. Okay, this is one of the first way of controlling the tryptophan operon. This is how, that means through repressible protein. That is what is called repressor. That means a repressible operon to repressor protein. That means tryptophan is a co-repressor. Here, inactive enzyme, inactive protein in the first instant, what we call apoenzyme. Okay. Then in another case, I told you already, attenuation. This we have to speak at length. Attenuation, in the sense here, there is a leader sequence, what we call, these are structural genes, and upstream to the structural gene there is an attenuator region, and this sequence, what we call leader sequence, will transcribe into the mRNA at the 5' end, and this contains different genetic codes, and this particular part, what we call the leader fragment of the mRNA, again have 4 regions, 1, 2, 3, 4, and within the first region, it contains 2 consecutive tryptophan amino acids, that means tryptophan codons, whereas the translation codons will present between 1 to 2 regions. When the ribosome, this is in prokaryotes, the translation starts before the completion of transcription, what we call coupled transcription and translation. The same thing will happen here. RNA polymerase will transcribe and ribosome start translating. When this happens, that means when the ribosome start adding the polypeptide, that means adding amino acids vice to the genetic code. Simply, we knew that, we have discussed in translation. The tRNA, the respect to tRNA, that means the with respect to amino acid, they will come, and if a peptide bond, likewise it will extend the chain, what we call polypeptide chain elongation. But in leader sequence, there is 2 issues we have to focus. Formation of stem loops, what we call hairpin loops. Which hairpin loop or the kind of hairpin loop determines the termination off, that means transcription off or on. Say, I told you there are 4 regions. The stem loop formed from 2, 3 is called as anti-terminator. It will terminate the transcription. I will tell you how. Then, whereas 3-4 termination, that means 3-4 hairpin loop is called termination signal. That means transcriptional termination is because of 3-4 stem loop, whereas anti-termination for transcription is because of 2-3 stem loop. And this is how they control the gene expression of lac operon. That is why it is also known as attenuator. Okay, the issue, I will tell you how it went because it is most important part. The rest of the compound is very simple. Just like if there is a co-repressor like tryptophan, it will prohibit, that's it. But attenuator is something like different. Say this is attenuator, what we call leader sequence. Then we got an mRNA. This is called leader region. Leader region. Okay. It have 4 parts, 4 regions, what we call 1, 2, 3 and 4. Within the first part there are 2 codons for tryptophan. Whereas between 1 to 2 we can expect termination signals, that means termination codons. Okay, this is the basic structure of a leader sequence formed from the attenuator, which are present upstream to the structural gene E. Okay, we will think like this. First, RNA polymerase has transcribed. Then it has, the mRNA synthesis has not been finished. It has been in process. I told you, coupled transcription. The ribosome will come and sit here. The ribosome will come and somewhere it sit here. Okay. The place of the ribosome, it start extending the polypeptide. Generally, the size of the leader sequence here is around 162 base pairs, and the size of the leader peptide is 14 amino acid peptide. Okay. Just imagine somewhere around 1 to 2, the ribosome has been placed, it has been sit, and it has been translating. You know what happens? I told you the termination codons present in between 1 to 2 regions. The termination codon comes, then the ribosome, the translation stops. What we call premature translation stopping. You know what happens? I told you already. Formation of stem loops. Stem loops is very important. Otherwise called hairpin loops. These are secondary structures. You know how it went? For example, since they sit at 1 and 2 region, it will not allow the second region of the leader mRNA region to form hairpin loop with third region. Here, different hairpin loops are formed. Hairpin loops are nothing but the complementary base pairing. But the end point, they form a loop-like structure because of non-pairing. Okay, like this. For example, this is a hairpin, and these are the pairing regions, and these are the unpaired loops, what we call. Okay, these are the paired regions. The second and 3 will form one kind of loop, and third and 3 will form another kind of loop. The thing is, since the ribosomal arrangement is such a way that it will not allow the second region to pair with third, the third region will pair with fourth, and forming termination loop, what we call, say for example, how it went. So this is a third loop, 3, this is the third region, this is 4 region, complementary base pairing, and this is a loop, 3, 4 stem loop, what we call terminator loop. Means it will not allow the transcription of EDCBA, the structural genes, because the mRNA that has been vehemently terminated because formation of loop. That is why 3-4 loop is called termination loop. Imagine this is one case. That means then there is a higher tryptophan. Then another case. That means what we call anti-termination. We require tryptophan. That means that availability of tryptophan is less. You know what happens? The second case. the mRNA, just say in the same manner, it will, when it comes to the tryptophan bases here, just focus on here, tryptophan bases, it will pass what we call stalling or pass. Why pass? Because there is no adequate availability of tryptophan. The respect to tRNA, if you have to search where the tryptophan is, so just like CCN through which it will have to come to the translational side, but it is not as speed as it is when there is ample availability of tryptophan. Here the case is different. So when the tRNA is in search of tryptophan, it's like pass, the what called stalling is blocked. The ribosome has been blocked at the tryptophan codons. This is very important. Tryptophan codons. That means this is second case, okay? For understanding, I am saying. Sorry, this is second case. Oh, okay. Blocked. You know what happens? When the ribosomes remain here, that means within the first zone, it will allow the second and third region to form loop. This is the second and third region to form loop. This is called anti-termination loop. Why? Because I told you 1 to 2 region, the termination codons contains between 1 to 2, and this has been involved in the loop formation. So it has been bypassing of the termination signals. So in the formation of loops, therefore allowing the gene expression EDCBA. That leads to synthesis of eventual synthesis of tryptophan by intermediate action. So the point is simple. If there is a high or adequate tryptophan, the ribosome if there is a formation of 3/4 stem loop, because the position of ribosome in such a way that the second region of the leader sequence in mRNA will not form base pairing with third region. That means 2-3 stem loop it will not form, but the third region paired with 4 forming 3/4 stem loop, and it will prevent the transcription, what we call termination of transcription, premature termination of transcription, when there is a high adequate availability of tryptophan. I mean, second case, when there is a less availability of tryptophan, the mRNA, that means the ribosome become blocked. That means there is a stalling or passing of ribosomes at the tryptophan codons present in the apex, what we call starting point of 5' end of mRNA, and because the tRNAs has to bring the respective amino acid with respect to tryptophan, during this process the second and third region will form loop, like 2-3 stem loop, which is called anti-termination. Second 2-3 anti-termination, 3/4 stem loop. So this leads to 2-3 anti-termination leads to on, whereas 3/4 termination leads to off. This is how the tryptophan operon is regulated based on the crux point of attenuator. Attenuator. This is very important. Attenuator mediated gene regulation.